Thank you very much, Nancy and Stuart and everyone else amongst the mining community that's been so supportive to our work over the last 15 years or so. I'm not entirely sure it's a symbiotic relationship as much as a parasitic one, because I'm not entirely sure you're getting a hell of a lot out of us getting in the way of your equipment and generally being annoying. But I have to say you guys have been incredibly patient and really generous in terms of assisting us get at the material, and we're often quite humbled when we turn up and the miners are actually getting so good at identifying this stuff and running out after the monitoring's going on that we're in danger of running out of a job in terms of the paleontology side of things. That's why I'm working on the DNA, because you can't do that, at least for the moment. Anyway, so, and thanks so much also for coming along tonight. Those of you that should be out there finding gold, I realise this is a bad time of year to be trying to ask you to come along to hear what we're doing with the bones that we keep taking away from you. But it's, of course, the time we've got to come and actually collect the material, because it's when you're most active. I think for those of the miners that can't make it tonight, Nancy is arranged to video this, and I think you are going to be forced to endure it at your AGM this year, which I apologise for in advance. So I'd just like to show you a couple of really important findings, I think, that we have been able to discover only with the remains that you guys are digging up. And I'm only going to talk about the genetics side of things, but I'm particularly going to be talking about how we can use the genetic information, the DNA from these ancient remains, to look at the impacts of environmental change. And this is the thing that I'm most interested in. The genetics really is just a way of seeing what happened in the past. And primarily, we're kind of interested in what the potential future will be if we keep changing the environment in the way which we're doing right now. So when I normally give a talk, I kind of put up these three areas. Just as a point of reference, the background to that slide is not Yukon, as you might have guessed. It's Cueva del Milodon. That's a terrible Spanish accent. I guess you all know that. In Australia and New Zealand, we don't have any Spanish, so we never get any practice. But Cave of the Milodon, which is the giant sloth. And you can see the giant sloth is right, there's a model of it right here. Oh, okay, awesome. And this is people, obviously, for scale. And this is a large cave in Patagonia, which is another place that we work. We go to all the coldest places in the world. And the reason we do that is because the cold preserves the remains of the animals very well, including the DNA with which we need to work. Now, I've put a whopping great set of arrows over there, so you can't really see it, but it's a lovely cave. It's enormous. It's a conglomerate cave at the edge of a glacial lake, and the wave action from the lake has eaten into the conglomerate and dropped down. You can see across the left-hand side there a big dike, effectively, but the cave itself is actually twice as big. Now, the floor is ground sloth, can I say shit here? Dung? I normally say shit, so as long as that's all right with people. Dung. Coprolites, whatever, shit. It's huge piles of shit. So it's about 2 meters deep in giant ground sloth dung. You can still pick up the big balls are still there on the floor. And the big dips and dents and holes you can see on it are very early archaeology, because they worked out very quickly that there were huge sheets of sloth skin, dried sloth, mummified sloth, had died in the cave and been preserved, because the dung acts as a dehydrating agent, had dried them all out. In fact, Darwin was showing a bullwhip that one of the local ranchers was using, which was about 1.5 centimetres thick of leather. Amazing material. So the archaeologists went in to try and find early humans and everything else, and they didn't backfill, which is an absolute no-no in archaeological terms now. And so you can see the original pits and for example, the trench here that someone's dug. These are massive early pits back in the early 1800s. Now, in the background, though, you can see quite a different kind of landscape, and that's when the local farmers worked out that there was a lot of money in these Megafauna remains that were in the caves, and museums would pay you quite a bit of money for it, but it was a real pain in the ass to dig them up with this archaeological technique, so they just used dynamite and shoved a whole bunch of dynamite in the ground and just blew it all up, so all the bones would come to the surface, which is a far more clever technique, but unfortunately completely destroys the stratigraphy. So that's why the back of that looks like World War I, because it is, in fact. Anyway, so we go to places like this and we get bones. There were some humans here about 5,000 years ago up against the far wall. There was a few human burials, but largely it was megafauna. And what we're looking to do is try and get the genetic information out from the genomes. That's all of the DNA in an individual. And I do a lot of work, I'm not going to talk about it today, on the microbiome. That's all the bacteria that is present in an organism. And that's turning into a whole huge new field of massive implications for our health, but also for evolution and the like. And so, you know, you've probably heard this by now, but it's still surprising to some people that the thing that is sitting in your chair, like you, is only about 10% you and 90% bacteria, if you count the number of cells or the number of genes. And you are walking around as a massive ecosystem with the bacteria largely running things. Which we tend to forget about. We tend to think of ourselves as human. And so there's huge impacts in there when you start taking antibiotics. I mean, bacteria are controlling your mood. Well, not controlling, but influencing how you feel. They're really important now in a number of diseases. And so we're trying to work out how the bacteria have changed over time as we start living in houses and sitting on our behinds for most of the time and eating large amounts of carbohydrates. So that's a huge area of research now, and how the environment plays into that. I'm not going to talk about that tonight. I'm just going to talk specifically about environmental change and genetics and how we can study this. So the reason that the Yukon is so good, apart from the fact that it's got cold preserved material here, is that it's got a lot of material from what we call the Pleistocene. So that's the geological, for those of you that aren't in the mining community, that's the last 2 million years. Roughly speaking, otherwise known as the Ice Ages. And you can see why when you look at this plot. This is oxygen isotopes through time, and they're showing temperature. You can use the oxygen isotope to determine at what temperature the water, which is present in ice in this case, was formed with, as a sea temperature or otherwise. And so across the bottom, this is time in thousands of years in the middle and millions of years towards the left. So you're going back 3 million years. That's when the Pleistocene starts off, and you start seeing these massive oscillations. It's an exponential scale, right, across the bottom. So it's a bit confusing because this bit has all been stretched out. That's only 10,000 years. You've then got the last part of the Pleistocene just showing you these massive huge changes in terms of warming at the top and cold at the bottom. And then you can look right back 2 million years and you can see it's been going on and getting worse right the way through that period of time. It starts off with the 41,000 year cycle. This is to do with how the planet wobbles, basically, and then really gets going into 100K. And the reason I'm showing this is because people talk about climate change now and we're really worried about it, although the debate is largely about CO2 and whether we're doing it or not, which is really not the point. It's important, but it's not the point. What we're really looking at is climate changes in this part here. That's what we're talking about, because those last 10,000 years, this bit, that's everything that you know and value about humanity, right? So farming, housing, societies, art, everything else, well, not entirely, because art does get going a little bit earlier, but that's everything we know about civilization is in here. Prior to 10,000 years, you got the hunter-gatherers. And one of the main theories for that is that until you had the climatic stability of the Holocene, this flat line, effectively, you couldn't farm. You can't do crops. You can't do big societies. And so why? Why does the climate suddenly stop this massive bipolar warm, cold effect and become constant? You know, even something you might have heard about, this little ice age, which caused, you know, the Thames to freeze over and death all over Europe. That's this little wobble here, right? That's how much temperature change you're talking about for that event, versus this kind of thing, which was the norm for the last 500,000 years. And so you look at that and you think, hold on, this constant temperature of our last 10,000 years, A, we shouldn't be trying to change that, because that's really good compared to the normal system, which is that. And if we're going to perturb the climate, the odds, if you look at that record, are that we're going back to this situation. We're not going to make that more prolonged. That's clearly an anomaly, this last 10,000 years. Why the temperature is so stable is a really good question. But basically what happens beforehand is normal. And Also, what happened? What happens when you do these massive climatic shifts? Can we see that in these ancient remains, these ancient animals? Because if we return to this, what's going to happen to us or the environment? And even if we're not going to return to it, it's just kind of interesting to see how things have evolved during that period of time, because the genetics of the animals and plants around us, they all evolved when the climate was doing this huge changes in climatic shifts, not in the last 10,000 years. So if you plot CO2, just because that's the one that everyone gets really het up about, you can see the same impact. So now I've got a linear scale across the bottom, 600,000 years, roughly. And the warming bits are the yellow, and the glacials are in blue. And you can see the last big glacial. I'm just trying to get my mouse onto the screen, but it's not. There we go. Way, found them. So there's the last glacial maximum, or the LGM, as we refer to it, is right here. It's about 20,000 years ago. And that's when the glaciers were biggest, when it was supposedly roughly coldest. Now, we are living in the Holocene here, and you can see that it's just one of a series of interglacials, these yellow warm periods. Now, I've plotted CO2, because I just like to scare the students with it, because it shows just what the levels we're now experiencing are like compared to the last, well, basically the entire Pleistocene. I'm just showing you 600,000 years here, but you can go right the way back 2 million, because we are now off scale, right? So last year we crossed 400 parts per million CO2, which was about, I'm showing the mouse on the screen, but of course it can't come up. It's actually in the black curtain at the top there, roughly speaking. So if anyone gives you the story that CO2 levels now are not particularly unusual, and that previous climatic records show that CO2 can do these kind of things, yes, it's true, but you've got to go back to the Paleocene, you know, and just after the dinosaurs are around, to try and find similar CO2 levels. This is all the previous ice ages. And what's happening is the CO2 is just following the degassing, the warming up of the Siberian permafrost in particular, but also here. And the CO2 is just showing you what warming looked like. I just want to show you, well, as I show my students, look, that's normal for the last glacial, last 600,000 years. This is where we are now, and we're heading straight up, which is going to be a really interesting experiment. But I'm not entirely sure we want to see the answer. Anyway, so if environmental change is happening all around us, what can we see about the impacts of those environmental changes, particularly in the DNA, because that's what we measure. Now, this is reasonably serious stuff. It's got big implications, and I'll give you a couple of examples from Australia, or Australia, which is not where I'm from, but where we're working at the moment. And the one you probably heard about is the Tasmanian devil, which is rapidly heading towards extinction, despite a lot of efforts to try and save it, because it has a facial cancer which it can pass to other individuals. Tasmanian devils largely communicate by biting, whether they're saying hello, or whether they're fighting, or whether they're mating, or whether they're just bored. They bite each other. And it turns out that this cancer is passed during that biting to other individuals. And you can see a fairly horrible, massive tumour on the front of that guy. That's what they look like, normally worse. It's a very horrible way to die. We don't know where the cancer came from. There's one other example that we know of, a rare form in dogs. They can also transmit cancer from one individual to another. Fortunately, we can't do that, and no one else seems to be able to. The point is that they pass this cancer to each other because genetically or genomically they are so close, they're so identical, they have no diversity. Can't recognize the cell being given to them by another individual as different from them. They're so genetically similar. You could take out an organ from one Tasmanian devil and put it into another one, and it would work fine. You don't have to do any of the tissue transplant stuff that we have to do. And that's the problem. They've got so little genetic diversity, they can't fight this disease. Similarly, koalas. Not one of my favorite animals, but they are high profile because they're furry and they've got big eyes, and Japanese tourists in particular like them. Now, they're all dying of chlamydia, which, you know, you think, but it's not actually any big deal. We've all got chlamydia as well. It's just, it's all part of your natural microbiome, getting back to that. What the koala really does have is a retrovirus, this thing here. And that's a virus that writes itself into your genome. It's like a computer virus, actually, same kind of thing. Very similar. You can pass it between individuals, And when you catch it, not only does it infect your body, it jumps into your genome as well. And it's picked up this retrovirus in the last 100, 150 years. We're not sure what from. I'd put money on something the Europeans have brought in, dogs or something else like that. And the retrovirus makes your immune system collapse, kind of like HIV. And so chlamydia, which they've all got, and as I say, most of the audience here has got, I'm not making any aspersions, but it's just part of your background, that then jumps out when your immune system is depressed. And so that's why they're all dying of chlamydia. And then lastly, wombats. Wombats are one of my least favourite animals because they just don't look very good on the whole. I'm a New Zealander and all the Australian animals don't really look that smart to me. But this one, if you are going to die, if you are going to become extinct, choose a better disease than mange. You know, of all the diseases to go extinct with, mange is really not high on the list. And that's just, it's an infection of lice, effectively, or scratching insects. And that's coming from dogs as well. And in each case of the koala and the wombat, I should have mentioned, sorry, neither of those groups has genetic diversity either. They're facing the same problem. None of those animals, and in fact most of the Australian species, as far as we can tell, have any genetic diversity. And as a result, any one disease just goes whoomp through the landscape. And why is the immediate question, because if you're going to try and save these things, you've got to work out why this is happening. Well, Australia is not a very nice place, and just in general. And in particularly, this brown bit you can see on this relief map is largely moving sand dunes every time you have a glacial. So in the ice ages, or at the height of glaciation, the thing we all think about is cold and the fact that that is bad. What we really should be thinking about, cold's not too bad unless you're under ice, it's actually the dryness. Because so much water is being locked up in the polar caps and in the ice mass, it's the dryness of the atmosphere and the water in the environment, that's what's killing everything. There's no water around. And in the case of Australia, what that does is mobilize all that brown area into just shifting sand dunes. It's like Mad Max without Mel Gibson. And you can't grow plants, you can't have any animals. The whole thing just becomes one big inhospitable sandpit. And all the animals are kind of clinging on for dear life at the very edges. And that's when their genetic diversity is completely reduced because they're tiny little surviving populations. And it just shows you, Australia's an extreme example of what these climatic shifts can really do to the environment or to individual species. It can knock them, they're surviving, but they're only just. So what my group does, or what I've always been interested in, is to try and go back in time in various parts of the world to see what the impacts of these climatic shifts are. If we're going to try and understand how things have evolved. It's not just living on the landscape and having more offspring than your neighbour or having slightly better mutations than your neighbour. It's much more, in my opinion, and I think increasingly we recognise this, it's how you adapt with these massive environmental shifts. One thing I should have said back here and didn't, was that this change is incredibly fast between the cold and warm. So that change between the LGM and the Holocene, we knew from ice records it was less than 50 years. Okay, that's a 10 degrees centigrade average change at the poles, which of course are extreme, but nevertheless still big, in less than 50 years. If you try and get your head around that, it's about 2 to 3 degrees at mid-latitudes where we're really concerned about agriculture. So a 2 to 3 degrees centigrade change we now hold as being unsustainable for the current economy. You couldn't grow things where you grow them now, and you couldn't run our crops. Now, recent work with ice core drilling in Greenland, where they're actually taking the core straight out of the ground, out of the ice, into a mass spec and various other machines. So actually there's no transfer, they're actually melting real time the layers of ice coming up. That suggests that this transition from that cold to that warm might be less than 12 years. 2 phases of 5 with a little bit of a break in between. Now, just get your head around that, 10 degrees centigrade change in less than 12 years. So that is an extreme. I mean, most of these other switches, as you go from cold to warm, are slightly longer, but you're still talking decades, 10 to 20 years. So when the climate goes, whatever drives it, it goes very, very fast indeed, at human speed. And we're always used to thinking of geology and the never, never. You know, yeah, sure, the planet's going to explode in 3 million years or whatever it is, who cares? But this is now, and this is really, really quick. And decades is something we should be able to get our heads around, because a lot of us would see this if what we're doing right now is going to have some of the effects that we're worried about. We don't know what the effects are going to be, but I put money on the fact they're not going to be good. So places to go to try and get records where we can understand what has happened in the past and what might happen now. Here's this, where I'm going after this trip in the Yukon, is to go to Wyoming, where we've got an amazing site, a natural trap cave. It's a 100-foot vertical drop, hole about from here to the wall, that is beautifully concealed. It's just a flat limestone ridge, gently sloping, overlooking the Bighorn Reservoir, and just slopes more and more as you're walking across it, until suddenly at the last moment just dips, and you can't see the hole until even walking, and I've done a lot of caving, you're walking up to it and you're taking a step backwards because it's like, whoa. And at the bottom of this hole, there are huge piles of large herbivores, so bison, horse, sheep, and even more carnivores. So lions, the big American lion, the cheetah, the American cheetah, presumably all of them with a surprised look on their face, just splat straight onto the ground. And you know, they're amazing animals. The cheetah, you probably wouldn't have heard about because it doesn't get any press over here, but North America had a cheetah. I don't think it was in the Yukon that we know of. It's not the African cheetah, although it looks exactly the same. You look at the head and the bones and the back. It's got the big air scoop, you know, like the V8 nose, so it can take air in at high speed when it's running, and the spine super bendy, and the rear legs come like 50% in front of the front legs, so it can run really fast. It's a puma. It's nothing more than a puma on steroids. It's completely evolved into a cheetah from a standard American puma. Quite remarkable evolution. And the lion there, this is about 20% bigger than an African lion. That you did have running around in the Yukon. So these are all incredible animals. They're a landscape that's just completely disappeared that most people don't even know about. Other caves we go to look at environmental change include ones where humans are hanging out. So this is a site I'll be going to in a couple of months. We've been working for a while. It's the world's oldest winery, which has got nothing to do with why we're working there. But it's about 5,500 years old in Armenia. And they've got all these buried huge jars in which they are making the wine, but also storing wheat and barley, human feces on occasion, which is often quite fun. And various other things. It's like a warehouse from halfway through the farming period, all beautifully preserved. To give you an idea of the preservation, that's a shoe they found in there. It's the world's oldest shoe, 5,500 years old, out of leather. Nicely stuffed with straw, because whoever was leaving it there didn't want it to dry. You know, if you've got leather and you let it dry with a crease on it, you never get it to open out again without ripping it or having crease in. So that's how you stored your shoes. But 5,500 years ago, it doesn't look far off a moccasin today. So we're working there to try and work out how the crops have adapted to environmental change, how the wheat and the barley dealt with all these big shifts that we're seeing. We also go up to the North Slope of Alaska. You guys are all very familiar with that, and bits of mammoth. And just earlier on this year, I was down in Antarctica, which is the first time I've been down there. That was pretty cool. We were drilling holes in the glaciers to measure changes in climate and also how the glacier melted in response to those changes, because we're trying to work out what caused the sea level to jump up every time it gets warming. Because when it jumps, it really jumps. You're talking about 50 meters, I think it is, or more after the LGM, and trying to work out which bit of ice, Greenland, West Antarctica or East Antarctica, melts is really important, because we need to monitor that now to try and work out which bit's going to fall over. And I put this in there just because you'd recognize this is Ken Boric Air, so it doesn't matter where you go in the world, they're down there flying us out to our field sites, which is, they do a great job. That's where that site was, just next to the Weddell Sea, and so we're measuring climate change right there and the melting. So how do we do it? Well, the main problem we've got is to try and detect the tiny amounts of DNA that survive in this ancient material from all the DNA that is in the environment today. So as an example, just while you've been sitting in the audience now, there's a pool of your own DNA that you've created around you, just breathing out. You're breathing out cells, you're shedding bits of skin and hair. Right around you now is a big pile of that DNA. And if we try and do work on these ancient specimens, the DNA that we're carrying in our sweat that we breathe out would completely contaminate them. So this is the kind of gear we have to wear, very fashionable, when we're in the lab. And I'll show you a video of how we do the work. The actual lab itself is all positive air pressure to try and keep out pollen, dust. We go in the mornings, downstairs. This is the state herbarium, so it's about a quarter of an hour from the university to try and hide from any genetic work there. We have lab clothes that we put on, and then we come into the lab and put on all the body suits and the breathing masks and all the gear. Everything that goes into the lab goes through an ultraviolet oven here. We just fry off the outside, because we worked out very quickly that anything that came from a warehouse comes with mouse urine and pigeon droppings and DNA on the outside of all the cardboard boxes and everything else. So we have to destroy all that and then ultraviolet fry the outside of everything to clean. And then coming through from the back of the lab, through this air shower into the front. And I'll show you that in operation. It's all positive air pressure. They're allowed to go into that environment if they've been to the university that day, because they'll be carrying DNA from all the modern experiments. So if I can do a video, I'll show you what it looks like to work in there. Here we go. So here's our lab manager, Jeremy, or was. He's now our deputy head. He's in charge of forensics. He does all the forensics work. Kitting up for work. So there's the bodysuit, the breathing mask, putting on the boots. He's about to put on the face shield and then the surgeon's gloves. Everything's been bleached and cleaned repeatedly to try and sterilize it. And so the surgeon's gloves go right the way up the arm, and that's to stop your bodysuit moving up and down your cuffs as you're moving around during the day. Otherwise, the cuffs will actually just be scratching all the DNA off your skin and dropping it into your experiments. So he's just about to put that on. In the background, there's the ultraviolet oven at the back right. There's the door. Everything just gets slid through on a processor. It's 2 minutes of high-intensity ultraviolet, and then it pops out the other side of the clean lab. There goes the surgeon gloves, and then you'll wear extra gloves on top of that for working. They're now up to 3 pairs, which I think is getting a bit paranoid, but they insist it's all necessary. There's the visor to stop the DNA coming off your eyelashes. Eyelashes are a problem. You're constantly moving them around, throwing stuff off. And then to see inside the lab, oh no, I might go right onto it. We'll see. On the inside, there's the air shower, which is taking off any dust which has been created by the ultraviolet oven. And everybody's invented their own little dances to try and expose all bits of their body to the air which is coming up. And then that's the actual main lab. And then each of these little glass boxes is a separate room with still air. So that's the human room. So that's where we work on Neanderthals. And also you can see the rose garden, the botanic gardens out behind us there. So the tourists have worked out they can see in the window and watch all these people in these white bodysuits moving very slowly around, because you're trying not to create air currents that will cause contamination. So it looks like some sort of weird zombies going on. So I think we're in a lot of videos of people wondering what science experiments are going on. So that's where we do the work, and then we extract DNA mostly from bone and teeth. That's largely what we work on. We do a lot of soils as well now. But that's a close-up of a woolly rhino from London, from Lloyd's Bank. Whenever they put a building into London, they discover megafauna, because, of course, the Thames is an old floodplain, not that dissimilar from up here. And that's here's London in 1960 when it was black and white. And you can see that's the Lloyd's Bank. New Zealand House, incidentally, got a lion and various other things. But you can see the actual cells still preserved in the bone. That's a specimen I've preserved in 30,000 years old. This big thing here is the blood and the nerves through the bone, and these are the bone cells themselves. And the little black dots are things we call osteoclasts. They secrete the calcium or reabsorb it according to whether you're stressing your bone or not. This is why if you go into space, if you're an astronaut, and you come back down to ground, they have to pick you up and carry you away, is because these little cells have said there's no stress. So grab all that calcium back, this bone is no longer being needed, use it for something else, and they get all floppy. Apart from the Russian ones, who always just get up and walk off. Don't have the same occupational safety and health. So how do we get the DNA out? Well, we find a bone. This is a moa bone from New Zealand. The Kiwis will recognise this. Try and identify it to make sure it's one you want to work on. You cut a hole into it. So here's a chunk of bone that I've taken out. We dissolve it in EDTA, which pulls the calcium out of the bone. It binds divalent cations, 2+. And then we separate the digest with proteinase, the cells, and separate the DNA just by binding it to silica. Works easily. And then we amplify it using various techniques you would have seen on any forensic show where you use enzymes to amplify up the DNA. So, to get to the Yukon, if we take these specimens and we extract the DNA, what can we see? So, one of the key things we're looking at is the impact of Beringia, the land bridge between Russia and America back during the Ice Ages. The artist's impression looks something like that, except I don't think all the animals got together for posing situations like that often. But, you know, that's what you're talking about. You've got mammoth, you've got horse, you've got muskox, there's a few carnivores hanging around. That's the general conception of what it looks like. What we're very interested in is what happens when you create a crossroads between the old world, Eurasia, and the new world, the Americas. And all the animals can run backwards and forwards with the people as well. And so what we do is we came up to the Yukon, this is about 2000, and we started asking around whether we could get some bone samples. And one of the first miners I came across was the Johnson brothers, Bern and Rom, up at Irish Gulch, who were very generous. They were monitoring up there at the time, and you can see, like many of the miners now today, a pile of megafaunal bones being used as a counterweight in the monitor. Ron did a degree, I think, in zoology, so was very good at running in and grabbing stuff and identifying it. Sorry, Bern, that's Bern, my bad. Sorry, Bern. And they were so good that, you know, we were announced that we were on the way, and this is behind the generator shed where Bern had put this together. I think it's something like 2 weeks when we told them that we were coming. And that is, every one of those bones is a DNA source. Different dates, different species, most of those are bison, as you can tell. Huge amounts of material. The mining community will be fairly familiar with that, but if I try and show this to people outside the Yukon, people have no concept that this is the kind of volume of material that is being produced per mine in a few weeks. In fact, some of the material I started working on was actually collected by Dick Harrington, and I just got an ID on this tonight from Chuck McDougall's place. I knew it was 60 Mile, but I didn't know who it was. Dick told me at the time. And that's, again, the sheer scale. Apparently Chuck had actually separated all the bones out into different types before Dick even turned up, which he was most impressed with. All of that, each specimen is a whole genetic resource about that animal and about that species. So to show what we can do with it, when you've got that many well-preserved specimens, I'd like to talk a little bit about bison, because that's the group that we do most of our work with. Now, this is a group that is common, I call it the Pleistocene cockroaches, because they are everywhere that you don't have ice, you get bison. Other species are a bit more choosy. And they range all the way from Europe, here you can see Spain and England, this is during the Glacial, all the way across Siberia, all the way over the land bridge, right the way down to Mexico. The bison went. Most of the specimens we collect were in this dark red area here, but the penetration went all the way down. And so there's huge amounts of variation in the bison across that range. You get changes in the horn shapes, whether they go up, whether they go back. These are the ones from the Yukon, the northern form. These are the ones from the lower 48 states. These are the Texas guys, the massive ladder-fronds. I'll show you a photo of that. That's Bess Shapiro, one of my PhD students. That's the bison ladder-fronds from the lower 48 to give you an idea of the sheer size. Remember, you've got to put the horn sheath on top of that horn base. So the end of that horn is going to be up around here. We're probably going to curl back in, but still, this is massive. This is like 6 feet, 6.5 feet. A really amazingly large beast down there. And so all of these different forms have been described as different species. And many PhDs have set out to try and use these as chronomarkers, where if you find a bison and you look at the shape of it, you can tell how old it is, and therefore the stratigraphy. You could date the layer of soil you're in based on the shape of the skull. And all those PhDs failed because they could never find any relationship through time or space that explained all these different shifts. So we set out to try and make sense of this by getting DNA from as many bison as we could, and particularly using the Yukon forms. And this is what we found. It was kind of a bit surprising. So what I'm going to show you is a big family tree of all the different bison sequences that we got from Russia and from China, mostly from here, but then down into the lower 48 states. And I'm going to color code them according to those colors up on the map, right? We've got, I don't know why we chose green for Russia, but whatever. Alaska, we find several different types, and then everything down in the lower 48 is relatively similar, and we've colored it blue. And so here you can see what we can find today in bison. If you go to a plains bison or a woods bison in the lower 48 states, that's all the diversity, plus all the ancient ones we can find in those areas too, for that matter. Very little genetic diversity. They're closely related to one another. As soon as you start putting in the ancient forms, we did 500 ancient specimens. We carbon dated 180 of them, and carbon dates aren't cheap. They're about $500 a go. So that's a fairly big bill, you can calculate. But we need to put carbon dates on if we're going to understand how these things are changing through time, how the DNA is changing through time, how climate might be controlling that. This is what we found as soon as we started dropping in specimens from the Yukon, in particular, and Alaska, and Russia. All of this diversity here, clades just mean groups, a group of related DNA. So we get one big clade, which has the modern bison as a tiny surviving remnant, and another big clade, and all of that is now gone. That was all the bison diversity that were here in the ice ages, that was here in the ice ages. And this is what survives. And that's pretty, and the scale across the bottom here is genetic diversity, how different they are from each other, how big the population is. So you can see these guys are incredibly diverse, whereas all the modern bison have got no diversity at all. They come through one little bottleneck here, where a tiny number of bison survived and went on to form the modern guys. All that is a massive population. I'll show you the size in a sec. The key thing, though, well, one of the funny things, anyway, we're working with the Russians, and all the fossils of bison, the oldest fossils, are in Russia. Bison evolved in Russia. The Russians always think of them as their animal that kind of got away. But if you look at the DNA, all of the Russian bison are here. They're a tiny subset of the diversity you've got in Alaska and the Yukon. In other words, the Russian bison have all come from here. All the ancient bison in Russia, for some reason, magically disappeared and were then reseeded from Alaska. I'll show you a map of how we think this has worked. How that happens, I've got no idea at all. How do you wipe out every bison from England to Siberia in one go? Well, we know their DNA doesn't get through. Doesn't mean to say they all die in one catastrophic event, but for some reason they all got killed. None of them had any descendants. And then Alaska just reseeded the world. Alaska, Yukon, I should say. So here's a picture of how we think this has happened through time. If we look at all these different DNA types from the bones, and we use the carbon dates, we see that bison definitely did evolve in Russia. You can see the fossils. They're back there beyond 200,000 years ago. And some point after that, they invade the New World. And we're still trying to get a date on that, but it's probably about 150 to 200,000 years ago. They come across the Bering Land Bridge and take over from horses as one of the big groups in the Americas. And so I'm showing a couple of the different types of DNA that we think must be present at that point. The Russian bison are in black because we've never got a DNA sequence out of them, so we just know they existed. We don't know what they look like. Some point between 200,000 years ago and about 80, which is our oldest samples, all Russian bison go extinct, shown by upside down dead Russian bison. And then a small part of the genetic diversity that exists in Alaska comes rushing back in and takes over all of the old world. So all Russian bison are yours, which all the Russian scientists we're working with were very upset about because they think of them as theirs. And so they still refuse to believe this, but it's quite unequivocal. Why? I've got no idea. But something really bad happened across Eurasia. And it's starting to see this in other species too, which is intriguing. For some reason, Alaska was not just a glacial refugia, it was the glacial refugia. And why is a really, really interesting question that we're only just starting to try and ask. Sometime between 60 and 20,000 years, you start seeing the bison coming down from the Yukon all the way into the lower 48 states. They are there before that point, but we just can't get any DNA out of them. So what we can see is, again, a subset of the Yukon diversity comes down. We pick that up in Natural Trap Cave. Eventually, the ice, it stops at 20 because the ice sheets are stopping any movement between this area and this area. But when the ice melts, you get the bison rushing together to have a big party in the Peace River area of Canada. And you see both types, the northern and the southern types mixing. But then it's like boxing, and then they go back to their individual corners. All the lower 48 bison head back there, and all the northern ones head back here. And basically it's forests, we think, across this zone, which kind of separate the two. So there was a period of mixing, and then they separate out. It's like, whoa, that was kind of strange. But if you look at the tree of all those different types of DNA, we can't see any relationship between what the skeleton looks like, whether the horns go up or they go backwards, or whether they're big or whether they're small, and where they fall on the tree, right? You'll have big ones next to small ones, and upward horns next to downward horns. All that shape and size doesn't seem to have any relationship genetically. And our conclusion, and we've got tons of different species supposedly on this tree, but they've all got the same DNA. They're not different species, they're one species. The one species that changes enormously in shape and size, which is kind of bizarre. That's just what I've said. So why? How are they changing in shape and size that fast? Well, I think it's climate again. It's the environment is changing, and these animals are responding very quickly in terms of the food that is available, sexual selection, where the females like big horns or small horns, which is kind of interesting. And then across space, different food in different places gives you different size bison. Now, you might ask, okay, how are you going to change things that fast? And I would agree, you don't normally expect to see an animal changing so rapidly enough that everyone thinks they're different species, when it's really one species. Well, the way you do it is this new thing we've discovered, not us personally, but the medics have really got all over this, a thing called epigenetics. And what this means is genetics that doesn't involve changing the DNA. You might think, okay, that's kind of strange because mutations and everything, that's all changing the DNA, right? Well, this is not. All this is doing is turning on and off genes. And as an example, you use epigenetics all the time. Actually, just what you ate tonight is changing some of your genes and what mood you're in and what season it's in. That's all epigenetic. Every one of your cells in your body has got the same DNA, same genome. And yet, each of those cells has done something completely different, whether it's a bone or a brain or a toe or whatever else. And the reason for that is epigenetics. You've turned on and off differing genes in each cell to make them become a bone or a nerve cell. And it's a very powerful system, this. We've only just started to work out that it's being used as much as it is. The prime example, a really good one, is women in World War II in Holland were, the population in general had nowhere near enough food. It was a really bad example of the Nazi occupation. The Dutch population was really well controlled. They had no resistance. And the Germans really exploited it. They starved them. And so women in World War II in Holland were very starved and had babies, as you might expect, that were very small, well underweight. And that was no big problem. But when those babies grew up and had babies, their babies were small. And I was like, okay, that's a bit odd. The mother didn't have food, and so she would have been stressed, but the kid had plenty of food. So why is the kid still small? Maybe it's because she was carrying that the DNA for the kid already, and it's been passed one generation somehow. The trouble is that second generation has now grown up and had babies, and they're still small. Okay, they're all underweight. So 3 generations on, you're seeing the babies from that population of mothers is still showing the signs of not enough food. And I don't think this is like a terrible thing. I think it's actually an adaptation. The mother has passed on a stress signal to the baby saying, don't try and grow big. there's no food around. If you try and be 6 foot, like most of the Dutch, you're going to die. There's not enough food. Bring it right back in and grow slowly. And it turns out that epigenetics is a key thing that mothers do with their babies. They can pass on information about their environment. This is one example. It's used all over the place. Plants use it all the time. But once we worked out that humans are doing it, that mothers and babies are doing it, we suddenly start realizing, hey, this must be real. And maybe it's happening all the time in evolution. You don't have to change your DNA to be small. You don't have to have a mutation that says be small, that's good. You can actually just turn off the genes. And that's what the mother does. The stress hormones actually pass a signal. And what happens is you get methyl groups, so carbon and 3 hydrogens, just the methyl group, stuck on the side of the DNA. And when an enzyme comes along to read the gene, if it sees these methyl groups, it just, nah, not doing it, just moving off somewhere else. And so you just put methylation along a gene, you can just turn it off. And you've got specific enzymes that can do that. So as I say, what you're eating, what season it is, you're constantly turning on and off genes to adapt. And the way you're doing it primarily is putting methylation signals along the DNA. So I started to think, is this the way in which these bison are adapting? If animals are having to change because the climate is going up and down so much that they haven't got time for Darwinian selection, you can't have a mutation turning up in one bison and then their kids being better than other bison, and then 15 generations from now you've got a hairier bison. You've got to do it now. You've got 10 years because it's cold, right? You haven't got time for 15 generations. You've got to instantly adapt. And this system, which the medics are really getting interested in because it's starting to turn up in cancer and various other things, I think is the way they're doing it. So what we did was we took one of Burn Johnson's bison from Irish Gulch, 26,000 years old, beautifully preserved, and we measured the epigenetic signals in the genome of this bison. This is the first time anyone had done this in the past. We did it right here. And we compared it to a modern cow and a mummified cow I had from a hut in New Zealand, because if you want mummified cows, New Zealand's a good place to go. There's cows everywhere. And what we did was we measured 2 types of DNA. A gene called a retrotransposon, which is like a virus that just hops throughout your genome. Most of our genome is this, it's junk. And you've got to try and turn that off. And so these black dots are showing you where methylation is stuck on the side of the DNA. And it's saying, don't read this virus, you don't want to go there. And that's what a modern cow looks like. And these are all the positions of the DNA showing that it's got methylation on it. Here's your mummified cow. This is from Takaka Hill for the Kiwis. And here's the Irish Gulch specimen, looking exactly the same. You can see that we can measure which genes are turned on and off, even back 26,000 years ago. This gene here is a single copy. It's a male gene, and it's to do with allowing male babies to be born. And you have a 50% on and 50% off, because the female's trying to turn it off all the time, the male's trying to turn it on. And so you can see the black dots are showing half of the sites we measured. There's a whole bunch of little boxes here. Half of them have methyl turned on, and half of them, the mother's part, had it turned off. So again, we can see exactly what genes are turned on and off. And we're now doing whole genomes like this from ancient bison, mainly from the Yukon, at different points in time to see how the animals are responding to climate by which genes are turning on and which genes are turning off, and whether climate causes them to do that or not. So that's pretty technical. Don't worry if it sounds pretty odd, because most of my colleagues are still trying to get their heads around this whole epigenetic area. If you've ever heard of Lamarck, you know, where the giraffes are growing longer necks because they're reaching for trees and therefore their kids have longer necks, it's basically saying Lamarck was pretty much right. Not for giraffes, but for us. And the scientific establishment hates it because it's anti all the simple things that we know. Anyway, so what we can do is we can calculate the size of ancient bison through time from the DNA. We can work out how diverse they are. And we can plot this through time. What we do is we say that the bigger a population, the more genetic diversity it has. And if it's really small, it has none. So by comparing all the different bones at different time points, here's all the carbon dates along the bottom. Those are all carbon dated bones. We can estimate the population size. So this is what we think. We think about 10 million bison are running around the Yukon and Alaska back at about 30,000 years ago. And you can see that population suddenly crash about 10,000 years ago. So this is the black line is showing population size. Here's 100,000 years. Here's the modern day. Bison are doing great up until here. And then crunch, crunch, down to only about 10,000 individuals. And that's the period of time where we get the climate change, the last glacial maximum coming in, which is what we think has done it. Humans don't turn up until this black line here, about 13,000 years ago. So the bison population has collapsed from 10 million to about 100,000 and then goes all the way down to 10,000. And there's no way humans are doing that anyway. Humans at that stage don't use horses. They haven't got bows and arrows. They've got wooden spears. And we reckon there's about 1,000 to 2,000 humans invading North America. If you've got 2,000 humans that can't move very fast and you've got to kill 10 million bison, no, it's just not going to work. There's no way you can do it. So this study, which we did largely of Yukon material, was the first one to show that climate was doing huge damage to these populations. It's not all humans. We're really bad and we do kill things, but in this case, someone else had done it before we got there. So I'll skip over, and you can't see this. This is the problem. The fossil record just shows you the bison, right? Lots of bison bones, still got bison bones, bison doing fine. It's only when you look at the DNA that you work out, hey, there was a huge population that went down to almost no population and then slowly recovered again. This scale is exponential, I should point out, from 100 million down to 10,000. You can't tell that from the fossil record. You can only tell it from the DNA. And that's the first time you start saying, hey, climate is really doing something not too good here. All right, I'm going to move through that. So we can plot key points at when climate does something. This is a study we're going to publish in about 3 weeks, and I'm hoping is going to get a whole bunch of attention because it says that warming, climatic warming, is the thing that kills everything, which, given that we're warming up the planet right now, is obviously not a good finding. And the way we've done it is we looked at as many different megafauna as we could find, all the different large animals that have been carbon dated, and we plotted their DNA and their carbon dates. And the way we did it, I'll give you an example here. Here's European bison. We've been measuring bison bones in Europe. And if you measure the DNA of any bone older than 52,000 years in Europe, you get this new species. We call it Bison X. I want to call it the Higgs bison, just because the Higgs boson was one of the big physics finds recently. And that was hard to find. This was completely hard to find. So I'm not sure if they'll let me do it, though. And then between 52,000 years and 29,000 years, you get your bison, the steppe bison. That's all you find across Europe, all over the place. Suddenly, at 29,000 years ago, you're back to the Bison X, this new species. And then only at the Holocene do you get the modern European bison just being created as if by divine intervention. Now, these changes, again, you can't see from the fossil record, but they allow us to say something happened here, and something happened here, and here, all of which was a major change to the landscape to actually wipe species out. And so we can measure these points when this species goes extinct, when that one invades, when it then goes extinct, and then the first one comes back again. And we're going to plot these blue arrows against climate. We do it with cave bears as well. I won't talk about that. And this is what we see. This is what we're going to publish in about 3 weeks. It's been accepted by science, and the editors are all excited about it. What I've done is I've just plotted across the bottom those oxygen records from Greenland. Okay, so here's modern day. Here's 60,000 years. And this is those changes in climate I was talking about before. The rapid warming shoots up and then cools down, then rapid warming again. So I've coloured all the warming bits in this kind of pinky colour. I'm not sure what you call that. Pinky tan colour. So those tan boxes are the warmings, right? And I've plotted them all. Here's the LGM, the last glacial maximum, the cold bit, and you can see that the temperature comes down and just sits quietly, cold, whoops, all the way through. Nice and stable. And then it starts warming up. This is the Bolling Allerod interstadial, named by some Scandinavian guy, Bolling and Allerod, I guess. It shoots up warm and then collapses, doesn't quite make it. And then we had another go, shoots up warm, and we finally get to the Holocene, and that starts getting stable. We've stopped the plot at 4,000 years. And so I've plotted all of the different species I could find. Someone's done DNA or radiocarbon about when they go extinct. And I've made two estimates of when they go extinct, the red and the black. Don't worry about the difference between them. What you can see instantly is the big cold period here doesn't have anyone going extinct. The extinctions all happen right on top of these rapid warming events. We call them interstadials, is the technical name. And these are the ones that take place within a decade, when suddenly you go from cold to warm, wham, in one go. And this is not what we think. If you look at the textbooks, if you read any of this stuff, it's all about cold. Glaciations are cold and they kill everything, because we don't like cold, so therefore it can't be good for anyone else. Cold must be doing the business, right? And yet what this plot shows, it's warming. It's only the warming events that cause extinctions. Cold causes species to retreat into little refugia, and no one's very happy, but no one goes extinct. It's the rapid warming, that change at the beginning of the warming period, that kills things. And so we've got mammoths and bears and horses, all of it goes extinct, population one after another, until you hit the cold, where everybody sits there and does nothing. And then as soon as it warms up again, look, here's your next lot, boom, 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 boom. And these, of course, are when humans move around and when they invade the new world. I should say that the blue colorings are the ones from the old world, and the black ones are from over here, the text in black. And so sure, when the Native Americans turn up at this point, the extinctions are occurring, and everyone's blamed that on the Native Americans, and quite possibly they were killing off the last ones. But I would argue it's actually the warming event that was taking place at the time that had caused all the damage. And that's why you don't see huge numbers of butchered bones all across the landscape. There weren't that many animals left at that point. If you didn't put humans there, they would have recovered probably and gone back to being a diverse ecosystem. Humans turn up at exactly the right point, when the populations are small and fragmented and really unhappy, and kill off the last few. So this is really worrying, because we're warming up flat out right now. What we can see from that data is that warming kills stuff, and it kills it very, very effectively. The way in which it does it, we're still trying to work out quite what it is about warming that's so bad. It may simply be that when you warm, it's the biggest change you see. From being cold to being warm is the biggest single shift. When you go from warm to cold again, it's always this slow decay. You go back into the cold state. It's the sudden warming which is the biggest single climate shift. Anyway, maybe I'll take some questions about that later on if you haven't followed some of it, because it's hard to get your head around there. But we think this is perhaps the biggest finding we've found from all the Yukon material so far, is that climate, not just humans, is responsible for all of those extinctions. So the last thing I'd like to talk about, just because it's relevant to this area, although we haven't got any samples of humans from this area, is how humans invade the New World. I'll just show you a couple of slides. And this is a study we've been doing primarily in Peru and Chile, because there's so many dead people down there. There's like skeletons just coming out of the ground, literally, depending on where you are, huge numbers. And so we sequenced the DNA from about 92 of the skeletons from down here, a few from Mexico, and a very old one we didn't do, had already been published. And we analyzed the DNA to try and find out what the genetic diversity was of the first Native Americans, and if possible, when they turned up and how they turned up. And what we've been able to do, this is the first time again this has been done, is using the DNA from those skeletons, we can estimate quite accurately when the populations in the Americas separated from Siberia. And that's this here. That's when we see the last time that human DNA in Russia is shared with human DNA in the Americas. So about 26,000 years ago. That's the last time we can see for sure that they were joined. And then if you look at the diversity of DNA in the Americas, we can see that the oldest diversity that we've got in any Native American group starts up around about 19,000 years ago. So in other words, that's when they break off from everybody else in Siberia. This is when we start seeing diversity that is only found in the Americas. So somewhere between 26 and 19,000 years, a group of people, we estimate about 2,000, were stuck, we think, on the Bering Land Bridge. And the reason for that is everyone else in Siberia buggered off south, because at this period of time, 26,000 years, you're heading into the glacial maximum. This is getting incredibly unpleasant. And so all the human population started moving down south. Anyone that was out here got stuck on this whopping great big ice along the Chukotka Peninsula, and effectively got stuck into the Alaska Yukon land bridge. They were stuck out here. We think they waited for about 3,000 to 10,000 years in this period before finally it warmed up enough that they were able to jump through the bottom of the Alaskan center, or perhaps all the way around the Aleutians, although that's quite a long route, and then move down this coast. We know they're coming down the coast because we can see the population size. This is what this blue plot is here at the bottom. That's from the DNA, how big we think the population is, right? So here they are stuck on the Bering Land Bridge, this flat bit, and at 16,000 years, the genetic diversity goes kaboom, just explodes. And what we're guessing, I think pretty safely, is that's the point when the population gets into the Americas and it just diversifies enormously. It goes up hugely in scale. So 16,000 years is when they come through, and we know that the ice-free corridor through here doesn't open to about 13. And so because the human population has just gone nuts at 16, and that route is not available for another 3,000 years, we think that proves that they actually come across the coast. And what you actually see is that at that point, the current in this area jumps up by 2 to 3 degrees centigrade, and all of the glaciers retreat. The tongues that are floating on the sea suddenly shoot backwards because of the warming water, and you get little chunks of land forming all the way along the coast. They're only small, but they'll be fine to act as stepping stones if you were interested in following seafood, seals, seabirds, and the like. And that's the human population coming shooting down in there. And of course, they get all the way to the bottom of Terra del Fuego by about 13,600 years. In other words, all of the Americas was covered in 1,500 years. People got in and went boom, from top to bottom. And the DNA, bizarrely enough, shows that once they're in place, having done that movement, they hardly move again. The DNA that we see now is very much as it was right back at 13,000 years. For some reason, you don't get populations moving around like you do in Europe. You don't get Romans coming through, or Attila the Hun, or the Mongols. For some reason, all the populations stay very, very static. So we're just trying to publish that now, but it's kind of intriguing to just be able to take skeletons from South America, not even from North America, because politics here is too complicated, but to be able to reconstruct the series of steps that led to the small group of people being pinched off in Beringia, eastern Beringia, and then finally being able to break out and take over the entire New World in the space of just 1,500 years. That means going through the tropics and all the way back out to the Arctic of Patagonia. So I'll close there. That's 3 studies, I guess, basically the bison, the impacts of climate on all the megafauna, and humans. That's a fairly small set of the kind of things that we're doing in our lab, but they all relate directly to here and kind of show what a simple bone in the ground, or a bunch of them, can actually tell you about all these events that we've been trying to reconstruct from archaeology, from history, from various other sources of information. This is tiny amounts of DNA. The techniques that we're using today, we can sequence the entire genomes of these ancient bones. That's only happened over the last year. So everything I'm talking about here is the last 10 years worth of research. The techniques have just changed completely in the last year or so. So the power of what we can address, every gene, every change is now available. And our main problem is trying to work out, well, what do we ask? Everything is possible. What's the most important thing to do? It's not cheap. One genome costs about $10,000. So on my budget, we're not going to do that many of them. But suddenly we can ask everything that you'd ever want. But only because you guys are actually giving us the material in the first place. So I'd like to close by saying thank you so much for sticking with some fairly high-tech genetics and deep time stuff. Particularly to Nancy and Stuart for getting me over here, insisting. It wasn't so much that I brought the family. Nancy told me I had to bring the family, or I was in deep trouble, having been here for so many years. But particularly to all of the miners out there, look, when we're running around, getting in the way of the cats, parking in the wrong spot, and generally being complete annoying prats, we owe you a lot of thanks for taking the time to be generating this kind of information, and we owe you a lot for that. So, thank you very much, and for staying awake at this time of night. I'm very keen to take any questions if anyone stayed with me through there. And if not, just make up something. I should have planted a question beforehand. Yo.
Just wondering why you think the warming period is so abrupt versus the cooling. What are your thoughts on that?
Really outside my expertise, but the climate people I talk to, it's something about the way the trigger goes off. The trigger is from a cold state to a warm state. A lot of people think it's the Atlantic conveyor belt getting turned on or off. And this is why we're doing a lot of work in Antarctica at the moment, is to try and understand ice sheet melting, because the way in which you do these big ocean current changes is largely by dumping fresh water on top of the salt water and shutting down the way in which salt water gets cold and sinks. If you can put fresh water on top, it can't sink, it won't sink. And that's what's happening, for example, right now off Iceland. You're seeing a slowdown, supposedly, in the rate at which the cold seawater falls and then retreats back underneath South America. So for some reason, something about that system seems to kick big when it goes warming, and then it goes through a series of false restarts until it collapses back into the cold state. And we're trying to work out from the Antarctic record who it is that's sending off that trigger, and the evidence seems to be it is Antarctica. Which part of Antarctica is a good question. It's a very contentious debate, but being from the southern hemisphere, we think it's Antarctica, not the Atlantic. And we've got a few theories about why warming might be so dramatic, but the overall, I guess, take-home message, we're probably wrong, is simply the system is on a knife edge, and when it goes, boy, does it go. It whacks into the opposite system and then decays slowly back again. And, a couple of thousand years to do the decay. And that's the bit. Why have we had 10,000 years when this is turned off? And by warming up the planet, I'm on that camp, you've got to bet that if we're going to do anything, we're going to go back into this bipolar state. We've had it for 2 million years. Something's gone wrong in the last 10,000, and we're just poking it with a stick until it starts up again. And, you can't predict that that's necessarily going to be good. So sorry, I don't know the answer to that. The climate guys have given me a couple of things. I don't believe any of them. I'm no expert. It doesn't sound reasonable. They don't really know.
Will you repeat me?
Yes, certainly. Sorry.
Do you believe, or do the other experts in the field of climate believe that it's the shock of temperature change that's obviously doing something epigenetically with those that's causing them to go into a pattern of extinction, or is it possibly creating an environment where bacteria and other diseases can flourish?
Yeah, good question. I think at the moment, we're probably only just having discovered that, in fact, it's the climate that's doing it in this way. Previously, people had suggested, well, maybe it's just getting cold, and that changes the vegetation, and then you get the swamp starting up where there's more water in the landscape and the muskeg and stuff, and that's bad, and then they finally go out. To actually say, look, it's these individual events that are happening is no one else knows that yet. It's only just coming out. So we haven't really got our heads around measuring those repeated experiments to see, okay, is it disease this time? Is it whatever? So we don't really have a good answer there. Disease is a real pain because I think it's an untestable hypothesis. If I measure your genome, I will find huge numbers of diseases in it because a lot of the viruses and stuff that you've caught during your lifetime have now been written into your genome. But when I measure that, it doesn't mean to say you died of that disease. In fact, it almost says you didn't because it had enough time to be replicated. And that's the problem with, I can find all sorts of nasty diseases in an ancient sample, but I've got no way of knowing whether that's what killed something. Koala would be a good example. So I don't think disease really, I wouldn't be surprised at all if it was contributing, for sure. But I think it's the magnitude of that shift and all the environmental changes that go with it. If you think of the changes you see even up here, one of the things that blew me away when I went to Alaska was, I said, I want to see the caribou, because we're up in the North Slope. He said, there's no caribou. So why no caribou? And he said, there's mosquitoes. Bad mosquito year, and bot flies. The caribou have all buggered off around this area. And you're thinking, how are we going to model that when we're trying to do climate change? Something such a small perturbation of one species annoying another species, causing these huge changes. It's not a very good example, but to me it really showed that all the modeling we're doing is going to completely fail to take into account simple on-the-ground interactions like that you'd never have, mosquito density as one of your climate model impacts. And so I think it's much more whole environmental change, and if you can change by that number of degrees, what we see around us now as variability is just nothing. And you'd have huge consequences of vegetation, snowpack, wind direction, God only knows what, El Nino years, otherwise there'd be massive things. Disease almost certainly would be in there, but I think it would be one of the horsemen of the apocalypse. I think you'd have all sorts. What's interesting though is that each species has chosen its own time and place to have the final crisis. It's not like one point in time where the climate was so bad that everybody did it. It's like it's a process that's going on all the time. And in many cases, like the bison in Europe, one bison goes out, instantly another bison species comes in. So the ecosystem overall is quite stable. Even though things are going extinct, they're being replaced. And I think maybe that's what humans have done at the end to disrupt the whole system, is that they've interrupted that replacement. Because if you can imagine climate change occurring in the Yukon and wiping out bison because all the grass went and it suddenly became fir trees, that bison from Alaska, which would normally start trying to push into this vacant area, all you need is a few humans in the area to take out the bold individuals that are coming across each year. Those are the ones you knock off. Suddenly the whole population says, well, I'm not going there, we're staying over here. And you've suddenly created an area without one species. And if that happens in a few spots, these gaps start to coalesce, and you can imagine small localized change becoming ecosystem-wide. And then you only need to take out a mammoth or something like that that has massive impacts on the landscape, and suddenly the whole thing just flops. Even bison have big impacts on moss and swamps, for example. You bring bison back, they cut up the moss very effectively and start introducing grassland. They're a fantastic landscape gardener, nowhere near as good as the mammoth is, but just changing those species, suddenly you can shift whole environments. And so I think that's how humans have caused such damage, is by stopping that stability, that replacement of problems with other species from neighboring areas. That's a guess, but that's how I think they might have done it. Yeah, so I hadn't seen you before, so how are you doing? Sorry, don't tell Bern about Ron. The evidence for the Polynesian connection is, is he there too? I'm damned, sorry. So the Polynesian connection, there are some similarities, and there's things like the log sown log planks and stuff like that, the way they construct canoes and some fish hooks and stuff. I don't believe a lot of that. I mean, some of the archaeologists I talk to say, look, you'll see that there's only certain numbers of ways to sew together planks or to make a fish hook. And while there are some surprising similarities, there's not a lot of convincing evidence. There's no genetic evidence. All the genetic evidence shows it's one group coming through from Russia, no Polynesian DNA at all. There's a population recently reported from Brazil on eastern coast with Polynesian DNA. And you're thinking, what? You go to America, you cross the Andes, and you end up in eastern Brazil. We do know that huge numbers of slaves were traded for the sugar industry from Polynesia, particularly from the Easter Islands and the like. So I'm very suspicious about the carbon dates of these ancient specimens. And when you find a Polynesian, what you're finding is post-Spanish slave trade rather than actual pre-Columbus contact. We've done a lot of work on that from chickens and various other animals. We can't see any sign of it. It might have happened. I really don't think it happened way up at the top corner of Alaska. If it did happen, it's going to be due east of Easter Island along that South American coast where the long distance canoes would naturally connect with currents, would take you right up to the northern part of South America. They're not going to take you all the way up to the Alaskan Panhandle unless you're really lost. So we can't see any evidence of it, and I think this is the problem of trying to interpret archaeology where people are really inventive and they're really clever and they'll often come up with the same idea. It doesn't mean to say that they learned it from one another. And trying to tell the difference between those two things is really tricky. And in no way do I disparage the Polynesian ability to get around, because those guys got everywhere. You guys probably haven't heard, but down where I am in New Zealand, we find Polynesian canoes, and certainly contact, on sub-Antarctic islands. They were right down on I think it's Auckland, it's either Auckland or Campbell, I forget which, hunting the mutton birds, the shearwaters. And you're thinking, you're in an open canoe next to Antarctica. I mean, I thought it was good enough you found New Zealand from Tahiti, but to go all the way down to Antarctica in a canoe? I mean, these guys are hardcore. So in no way do I doubt that they had the ability to get around. I just don't see any evidence that they really significantly influenced the cultures, either genetically or archaeologically. Not yet. Maybe, but I don't see it.
Wondering what your work is doing on the idea of natives coming here in waves. I know the Glacials have stories about people before them. They call the ancestors. So they were here, 12,000 years ago.
Yeah, yep.
I know there's some people who think that natives came over about 35,000 years ago. Some people say 50. How does your work relate to all that?
Yeah, we do. There's certainly waves, but there's three generally accepted. So the Eskimo, Inuit, latter peoples the last 1,000 years or so. The Nardine, famously the famous group there would be the Apache, who have actually been forced down. That's about 8,000 years. So they're a separate language group and a later arrival. But then everything else seems to come in that first wave. And that's the one that I consider decolonization of the Americas. And one of the giveaways is that you'll find the same set of DNA from the north of America to Patagonia, and then variants from it in each area. They went right the way through the country in one go. You then get subsequent waves coming in, but they're really small and localized, like the Nardine. So really, we see no genetic sign of anything before that group at 16,000. Certainly there's carbon dates and various other evidence that have been argued. I don't believe any of them prior to 15,000 years. And the fact that it's the same DNA set, which is exactly what you get. If you've got one population moving in and then rapidly expanding, every population that's formed has that same starting diversity. Whereas if you have separate waves, you'll get a whole bunch of different types down the bottom and then a different set at the top and otherwise. We've got homogeneity and then invention from that homogeneity in each place. So over the 10,000 years, the new lineages evolved, but the same starting pool exactly from north to south, which I think is the most convincing evidence. You've got one wave just boom, taking over the place. If there was anyone there beforehand, I've not seen any evidence, I believe, about it, but they had no genetic contribution afterwards. And that's hard to believe. If you're going to take over the Americas, you're going to have a decent population. There's food everywhere. You can't move for food. Why it would be a small population that's A, hard to see archaeologically, and B, could be swamped out by this movement at 13,000 years, you're doing something wrong. If you've got that much food around humans, throughout history, you give them free food, they explode, and they take over until they kill the place. So I really find it hard to believe that humans are here 30,000 years ago and didn't leave enormous signs, because it's megafauna, it's seafood, it's everything you want to eat. And that's just our record. We don't do things slowly. If we can breed, we breed. And we're like flies or rabbits or anything else. Just a slightly longer time scale and harder to kill.
So your evidence, of course, the DNA evidence, is that it's changing some things? Is there some reaction from other
Certainly the climate change megafauna one will get attention. I think the invasion at 16,000 years of one group into the Americas is not going to raise too much. We're basically refining what's generally accepted already. We're putting precise dates on it, but the pattern's pretty much accepted. The climate causing the megafauna to go extinct, that's going to cause a ruckus, because the general idea now is humans did it. Humans did it all over the world. They did it in different ways, but it's always humans. And while I don't deny that humans do the last bit, saying climate, particularly warmings, that's going to really, in 3 weeks we'll find out.
Just one last question. How, and if so actually, have you been able to apply your interest in the microbiome to the current research you're doing right now?
Yeah, well, we're actually sampling. I'm going to come back, Diana and co, I apologize in advance, and try and take some teeth out of the bison, because we've only been doing tooth roots so far. Because while, the logic connection here is that the bit that we work on is the calcified bacteria on teeth, the tartar from the dental advertisements on TV. And that's the only microbiome fossil that we've found. None other seems to exist. Even the fossilized poos aren't very good, because once they're laid, they undergo a whole bunch of decomposition, bugs fighting each other as the poo is cooling down. So before it gets totally frozen, there's enough action going on there that what you get now isn't what came out of the animal. It's big changes. Whereas the bacteria in your teeth, that's been calcified every night. Every night you go to sleep, that biofilm is calcified in place, which is why the tartar starts building up. And I could show you some lovely photos about that, but I won't. And so that is the only record we've got of the microbiome back through time. And we can follow sugar, when sugar comes in, and when farming comes in, we can see all these huge nasty changes, all of which we're convinced are associated with all diabetes and heart disease and all the things we've got now, I think are strongly connected to the bacteria. The animals, they're healthy. They eat rough food and not lots of processed flour, so they don't get that kind of pulp that forms around our mouths, around our teeth. The reason we've got so much of this bacteria, the tartar, is we eat this processed flour, and if you've ever noticed, your saliva dissolves it. It's been, we've chosen wheat to do this, and your amylase, which comes out of your saliva, actually starts digesting, if you're having bread or biscuits or cake or whatever. Even as you eat it, it's turning into a mush because you're starting to digest that stuff. It's that kind of white goo that sits around the bottom of your teeth, which is perfect bacteria food. It's like you're giving them this pre-digested sugar stuff, and they go nuts. And that's what's forming these huge deposits. Wild animals don't do that. Furthermore, they're eating a lot of silica and grit, and that's cleaning their teeth all the time. So that's the problem, is we can't get this tartar off anything else apart from domestic dogs, cats, and us. And what we're wondering, though, is in the teeth of the bison and the horse, you often get these sharp, they've got really sharp teeth with pits in between. We're trying to work out whether the bacteria might be being calcified in place at the bottom of those teeth. And so we're starting to sample whole teeth and try and get the microbiome out from there to try and do this. But at the moment, the problem is we don't have a fossil record, and no one does.
And they probably don't have caries or...
No, they've got fantastic teeth. Just like all our hunter-gatherer ancestors. Beautiful teeth, no health problems. Farming comes in and it just goes to hell. Sugar is a killer.
Do you have any examples of how the genetic material that you're gathering in Beringia is contributing to your work, to your studies? So that we kind of know what you're doing, what's happening with the material that you're gathering here?
Other than the bison and stuff, you mean? Or?
Yeah. Just what do you, I mean, because the bison is, it's just, you look at such a global impact. What's the material here? How's that contributing to things?
Well, it made up the large part of that Beringian story. So we got a bit from Russia and a bit from China. Most of it came from Alaska and Yukon, then a little bit from the 48 states. A lot of the work we're doing at the moment on the Beringian material is also looking at the connection between this area and the lower 48 states and how and when that occurs. So the ice sheet that blocks it off, there's a lot of question about how it happened, when it happened, and what the impact was on animals. And so we're trying to use the genetics to study the connectivity of here, presumably down through the Tintina Trench or whatever route they're going to use, down to the lower 48 states, trying to work out when that occurred and when it was possible, when it wasn't. And we're trying to do that by measuring lots of different species and comparing the difference between the two. Other than that, the epigenetics research, where we're looking at how change in response to environment might have done all these size changes, that's almost certainly going to be mostly here, because that's where the best remains of all the frozen material are. So principally that work will be here. Those would be the obvious examples, I guess. We're doing horses, bison, bear, lion, muskox, well, not so much muskox, brown bear, Arctidus. We're doing all those groups in this space, each one of which has got a different story. When they go extinct, how they move, and that's all telling you about what the environment looked like back at that stage. Karen, can you think of a better answer? No, all right, good enough. Yeah, already. Yeah. But basically, the Yukon is the large part of pretty much every study we're doing, because that's where the bulk of the materials come from. And, you know, Alaska has given us some stuff, but the miners there are far less approachable and far less interested, without a bulletproof vest anyway. And I'm not sure how much monitoring is going on over there anymore either. And the monitoring is the key. Without the monitoring, you get nothing. Well, almost nothing. You know, we've just seen that. We went out to 60 Mile yesterday and just seeing all the ripping that's going on. We're walking over site after site finding nothing. And you compare that to any of the monitoring operations over here, and it's just like, whoa, every day, huge new material. So since that in Alaska is not quite so common, we're not getting a lot of material out of there. So as far as, I think the Yukon's really amazing, and I really do hope you guys make a much stronger effort to build paleontology as a treasure, both for tourism, but just for natural resources. You're not going to get it anywhere else, certainly not on this kind of scale. And as far as the DNA goes, as far as the bone collection, you know, the bone collection the Yukon government is doing through the miners has built up a collection already, which I would say would be not the biggest in the world of megafauna, but up there. But within a year or two, if they keep on going the way the miners are generating material, this will be the biggest collection of Pleistocene animals anywhere in the world, will be in Whitehorse. And hopefully here, when they build the new paleontology centre. And that's pretty amazing. You know, there's a lot of people in a lot of parts of the world putting a lot of money into this, and you guys are going to completely blitz them because of the sheer amount of material coming in and out. And all those specimens can be used. I'm doing DNA, but you could do anything. Any question about climate change and evolution and anything else is going to be largely done here, principally because of the action of the mining community. So that's why I really reckon that, you know, Paleontology Week or whatever, there should be a much bigger thing here. Gold is great. You know, I'm not really that excited by gold, but you can go to many parts of but you can go to many parts of the world and see gold and gold mining. There's very few parts of the world you can go to and see megafauna. I mean, that's just not done, and no one's advertising it, and no one's really making a thing out of it. And yet, I would argue there's more interest in megafauna out there in the public watching documentaries or anything else than there is in gold. Gold's great, but you know, you've seen one gold shiny bit, you've seen them all. Whereas, you know, feel free to give me gold shiny bits and try and change my mind on that point. But at the moment, I don't really see it. Whereas, you know, you're finding mammoth tusks and bison and horses and completely weird animals and different bits of them. That stuff is completely unique to here. Now, not even Alaska, I think, is doing anywhere near that now. And so as a result, you've got this amazing material of scientific public value, which at the moment is not really being pushed that hard. But the collection that they're doing is the first step, and they're doing that really, really well. What you now need is a big centre where research is done on this stuff, where you can display this stuff. And as I've been saying to Nancy and Stuart, I'd like to see programs where tourists go out not to do gold mining, they go out to see megafauna. You know, because that you can't do anywhere else in the world. How you deal with the liability is another question, but you've got a huge resource here that is not being used in that way at all, that I think could and should be. I'm probably a bit biased on that, but.
I don't know the literature, the scientific literature that you're referring to. It's fascinating. I love your story. I know bits of the paleontology story from having lived here and speaking with people who do jewelry out of the mammoth.
Mm-hmm.
In the history, the literature that you've been spending time with, is there a clear distinction between Alaska and Yukon finds, or is it all kind of blended into this Alaskan?
It's pretty homogenized in the sense that the 60 Mile material gets lumped in with the Yukon, and yet effectively is Alaska or as near it, damn it. You know, I mean, no. I think it would have been if Alaska was still as active and contributing as much material, but they've kind of written themselves out of the equation. You guys are the only ones left standing in a number of ways. It would be, and they're not going to compete anyway. They're not interested in doing so, and they wouldn't be organised governmentally in a way that would facilitate it. It would be too much of a mess. Whereas here, you could really market. And someone was saying that, I can't remember who. I think it might have been Bruce. That really, you know, the symbol of the Yukon should be mammoth tusks, and that kind of exhibit should be the thing that you're pitching as the Yukon, as much as gold and the pickaxe. And there's no way Alaska is going to go anywhere near you on that one. Anyway, big market, I think. I think people underestimate just how many amateurs and kids are out there that want to see this stuff.
Alan, I think Sebastian had a question.
Sorry. Yeah.
I just want to know where I can find your paper once it's been published.
Oh, well, hopefully it's going to be all over the bloody newspapers, so it won't be difficult. But our webpage, we've got a webpage for the Australian Centre for Ancient DNA, has all the papers that we publish and a lot of videos and background stories and everything else. So it'll certainly be on there. But if not, our email addresses are on there, so just give me a yell on that. Or actually just talk to Nancy. But I'd like to send it up. In that paper, I'd like to point out, I acknowledge and thank all of the miners here that have contributed material, because you guys were key in that. So I'll send you all a copy and force you to read it at some point when you can't sleep.
Just don't test us on it.
Yeah, no, well, don't find any errors in it. That's more what I'm worried about. You guys have done extremely well. Anyone else?
I'm still puzzled by the, but you can't answer it, why the Holocene is so damn
Yeah.
perfect, eh?
Yeah. If I was religious, it would be a fairly easy answer. Yeah, why the Holocene? And it is, you know, humans have been around in Europe since 45,000 years, on the planet probably 100,000 years, and we never did anything, you know, not really, until the Holocene.
But you know, in that first graph you showed, it showed the Holocene on the right, and then it showed, I think, I don't know, 500,000 years in
Yep.
the middle.
Yeah, yep.
And then it showed so many million years on the other end.
Mm-hmm.
And you have the large up and down, you know, of the graph, right?
Yeah.
I wondered, well, would one of the in between, if you were to work, say, on the
Oh,
right
yep.
of your $500,000 period where you have
Yep.
the blip that crosses the Holocene line, would
Yep.
that time frame be
Here.
around 10,000 years?
It's possible that, and I've wondered that myself actually, and haven't gone back and actually calculated the length. But yes, almost certainly, a couple of those periods would have been almost that long. 10,000 years is not
We're probably right on.
exactly. We're towards the tail end of what has. I should know that answer. I apologise that I don't. But that record is all there. That's easy to calculate. All that information is right there. So how long, on average, or even at extreme, has one of these warm periods lasted? And I bet you it's not much more than 10, but it'll be up, you know, as you can see on that chunk there, that's a reasonable amount of time.
And yet it goes, in that time it goes above the temperature of the Holocene.
Yeah, it goes,
So, you know, yeah.
it peaks up. The CO2 doesn't, which is interesting, but the CO2 is generally following behind the warming. It's not driving it, which is, I think, how we've changed the situation now. We've made the CO2 so high that it's actually potentially starting to drive the equation, whereas it never has previously. It's never been high enough.
So what causes warming?
Well, again, a key question. We know that the periodicity of how it changes matches the wobble of the Earth as it goes around the sun. We don't know why that causes this bipolar impact, and therefore why it might stay warmer or longer in one period than it does in another. There's a lot of modelling going on. We thought we'd found it with some of this new data we've got on the animal study. We're putting out a paper suggesting, in particular, it's ice over the North Atlantic is an amplifier that as you turn on and off the Atlantic conveyor, you get huge amounts of ice, we think, from models forming out over the Labrador Sea, and that reflects a lot of light and really shuts down the conveyor belt as well. And we'll publish it. It's just the reviewers aren't as convinced as we thought we were. That it seems because of that exact placement and the currents, you rapidly get ice and you rapidly get collapse of ice. So it's almost like an amplifier system. If it's cold, it's really cold and reflecting light. As soon as it warms up, the thing turns off and you're back into heat again. And we are wondering if this is why that particular arrangement of the current across the Atlantic and the ice coming off the Labrador happens to be a switch, which is going on and off. And it so happens that the wobble of the Earth is exactly right for that latitude to have ice at its maximum when the Earth is dead upright. I always get this wrong. It's when the Earth is dead upright is the problem, because when the Earth is tilted over, even though it's colder in winter, the summer is hot enough to undo the cold growth. It's when the Earth is completely flat, so that winters aren't particularly cold, but summers aren't hot. That's when you get the biggest ice growth. And it so happens that where the Labrador Sea is, in combination with the Earth in that position and the current, is exactly right for maximum ice growth when it's cold and collapse when it's not. So we're putting that out as a model for what might have been a driver of those peaks.
And what about the elliptical?
The actual wobble around the Earth, that's, again, it's been related to the difference between this 41 and the 100K and why one might be dominant versus the other. But yes, they're definitely associated. How you convert an association into any sort of cause is the bit that I don't see any convincing evidence for yet, but that's well outside my area of expertise. But I haven't seen an easy, simple answer. I mean, that's the problem with all these Earth climate things. There's so many factors. You know, the Antarctic work, I was blown away when they told me that it's as simple as the current around Antarctica, the deep current, which spins off these cold plumes and controls fishery industries in Peru and stuff like that, is all dependent on the wind. And the wind freezes the ice on the sea, and as the ice freezes, salt is excreted, because as soon as you freeze water, anything in it comes out. The heavy salt brackish water sinks, and it sinks all around Antarctica when this happens, and because of the spin of the Earth, as it sinks, it starts to drag, and that's what drives the current. That system is beautiful, makes a very strong isolation, keeps all the cold in Antarctica, keeps the rest of us warm, until you put fresh water on top of it. So as soon as you melt a glacier, for some reason, a big one, and you put fresh water on top, it freezes, but no salt comes out, because it's fresh water. So it sits on top, freezes, no salt, current shuts down. So at that point, the current goes around and goes, woo, round the bit that's got the fresh water in it, because it's got no driver at that point. And that aneurysm in the circle sends the cold water off into a complete, formerly it was Peru, now it's Hawaii. Something as simple as where the fresh water comes out has such huge impacts on global climate. You start realizing that modeling this stuff and trying to work out why these things work is just a nightmare when something as small as the wind direction and where the ice forms changes whole current systems. And I keep telling the climate people I'm working with, you guys are doing a crap job on communicating these models to the public, because rather than whether it's CO2 doing it or whether we're doing CO2, it's this kind of thing about how the global climate works, the sheer mechanics, which are quite simple when you look at it. That's the bit that's so awe-inspiring about what we're trying to do and why it's so complex, trying to work out how Earth's wobbles can have these kind of impacts. This is the stuff that should be being taught in schools and should be taught to the public. Get away from the debate about the CO2. It's where it's got stuck, and it's really not helping anyone. Whereas these kind of basic physical principles, anyone can get their head around once it's explained to them. It's not, I don't understand. I was never trained in this stuff, and it's just totally simple when you see it, but complex in terms of how it would work.
Well, I think we're looking at CO2 because that's one contribution we're making. Other than that, we're not making a contribution.
Yeah, no, I don't in any way say that we shouldn't be looking at CO2, and I'm totally convinced we're doing it, and I'm totally convinced it's warming. It's a greenhouse gas. If you can prove how you can put CO2 in the atmosphere and not warm the planet up, you'll get a Nobel Prize, because you're undoing a whole bunch of physics really quite successfully. No, totally true. But I think the debate has got stalled over that. The anti-climate change guys have thrown enough crap into that people are just desensitized. What I would say is that what we should be being educated about is how the global climate works. Not the CO2 system, that's good, but this kind of changing currents, how wind direction or ice thickness, that kind of sheer mechanics about how the global climate, what the model is looking at, that's the stuff that should be advertised and educated, that we should be educated in. We should have the basics of how our global climate system works. And I would deny anyone in the audience here, with a couple of exceptions of the scientists, myself included, to have a first clue about global climate. I could walk around my entire building, a biology building, and ask anyone if they knew what the MODC is, which is the technical name for that current system. It's a meridional overturning current. And it's how all those cold currents go deep and then come up to the surface and where they go. So where all the currents are. No one in my building would even know what the MODC is. And we're biologists. You know, and I would argue that the public, same thing, not a clue, not a flicker. And that's the single basic, most important bit about heat transfer on the planet. And we don't know it, or you know, we're not told about it. So I really think they've not done a good job in that regard. They've had opposition, but still, I think we could be doing a lot better. Anyway, that's kind of a personal bias.
You're an excellent communicator. You did a wonderful
Thank you.
job on this presentation, but I think you've just explained also inadvertently in your little conversation here what the problem is. You know, like you didn't really describe the Mankiewicz type cycle that you avoided using that word.
Mm-hmm. Yeah, well, I mean,
but it's the same sort of thing.
I guess.
You know, like this is why people.
Yeah. We're trying to pitch it to the level of education of the audience, and
You've explained it, but without actually using the word.
yeah. And I agree, and I think that's where, though, the climate specialists should be starting up programs for those that are interested in learning more, can easily get that information. And they say we do, it's on the web and the like. I would like to see it much more integrated into school programs and NPR and stuff like that, stuff that you can actually listen to and pick this stuff up. And I agree, it is a problem that we're told all the time to speak at the level at which the audience is comfortable with, and too much media training, which is what a lot of them get, actually tends to lead to that effect, right? You kind of reinforce. You're not trying to explain difficult concepts. You do, but you kind of go around it without using the terminology. And that is a problem in the modern media world, as opposed to the good old days where we'd stand up here and say, you should all know this, and just assume that was right. The idea of knowledge for knowledge's sake was a good thing. Anyway, yeah, that is an issue, but still, if a whole biology department doesn't know the basics, there's a problem going on with science itself about communication. I should probably release you all. Yep. Sure.
But did I read something in the paper about Antarctica, the ice actually, like, it's not just one big massive rock, but it's actually
Two.
hollow in the center, and the ice goes way below sea level? So
Yes.
there's a possibility of seawater getting,
Yes.
you can have the ice just floating all of a sudden, like the whole sheet could just,
Exactly right.
so an ice cube water just melts.
Yeah, and that's again scared the crap out of me when the climate people started explaining this stuff, that a large part, even of the West Antarctic ice sheet where we are drilling, is under sea level. And it's actually up at the edges, holding the sea back, so that as you get a bit of melting and that like a dike on one of your dams starts going, you would logically get massive influx. It doesn't need to be much, it's like a meter or so. And you suddenly get huge influx into the interior of Antarctica of the ocean, and massive destabilization of the ice. And this is how they think that you do get such big sea level changes with these warmings, is the West Antarctic ice sheet in particular, this one here, is very liable to that. You don't need much collapse at the edge, and then the whole interior suddenly just goes whoosh and floods and melts And then all that water comes out. The West Antarctic ice sheet will give you about 2 meters of sea rise, roughly speaking, which in the greater scheme of things is relatively small in geological terms. In human terms, there goes all of Bangladesh, New York, every major harbour, most delta communities in the world, and all the religious fights going on around them. One go, bam. You know, that's the apocalypse in a can, and that's just the West Antarctic ice sheet. So we are very vulnerable to sea level change on that scale. You know, it doesn't take much for New York to be inundated with the next hurricane that comes along with a slight sea level rise. Not only the hurricane's going more north, getting closer to something like that, the sea level's also going up. So, you know, another half metre, and that whole thing is just going to go in one big insurance claim.
Has that happened before, that flooding?
In Antarctica, they're still trying to measure this, because this is key, is to trying to work out how previous phases of warming have destabilized Antarctica. That's part of what we're doing with that drilling. I don't know that well enough to give you a straight answer. I know that the models exactly predict that is what will happen. What we're trying to do with some of this drilling is determine whether the last warming phase 120,000 years ago, which is closest to where we are now, did that completely destabilize the West Antarctic ice sheet or not? The model at the moment suggests that it would. So the model suggests that at that point in time it did melt, and that contributed roughly 2 meters of sea level rise out of, Berto, how many meters went up in the LIG, the last interglacial? About 8. So we know 2 we think comes from the West Antarctic ice sheet. Where the other 6 are coming from is a really good question. So that even if you take out the West Antarctic ice sheet, where you see a nice natural way in which this will happen, people like Berto and co are trying to work out where the other 6 meters come from, because if we're warming it up right now, it's a really important knowledge to work out what happened last time, because we're monitoring West Antarctic ice sheets, but it's a good question where else it is. 6 meters is a lot. The East Antarctic ice sheet is really, really thick, and that if that even small chunks of it go, because of the depth below sea level and the depth above it, that's huge amounts of water there. The West Antarctic is relatively thin in comparison, but then you've got Greenland, and then you've got other places, and trying to work out who did it, which is what I'm trying to do with the DNA in the ice, is really key right now, because you want to be watching that bit really, really closely.
How far back are you looking in the ice?
Well, we've got last interglacial ice in this site. So on that series, as we get towards these mountains here, we have hit last interglacial ice, which already tells us the ice sheet didn't completely go. The question is, are we measuring ice up around a high mountain little isolated bit, or was it actually more significant? In which case, you know, we don't even get the 2 meters. 8 meters has got to come from somewhere else. It wasn't the West Antarctic ice sheet. So already we're pretty excited about what we're finding. And I'm trying to use the DNA in the ice to actually measure what the ocean currents are doing around Antarctica, because the ice is being, the snow is being blown on as sea, well, partly as sea spray, which forms snow and then it gets dumped. And so I'm trying to measure the microorganisms that are in the water that are getting blown onto the ice to try and see whether fresh water or salt water is being dumped into Antarctica. So that's what I'm doing with that series of, I don't know, we know there's microorganisms because when I filtered the cores, we had 2 to 3 meter cores, I stuck them through a filter while I was down there, so I didn't have to carry them back. And they're coming out green or brown or whatever. So there's definitely microorganisms in there. We're just doing the DNA work on that now to try and work out, can we see fresh versus salt and at what time periods? And I think we need to be doing more of this, where we take biology and start testing some of the geophysical models, because those guys have done great stuff, but it's all been physics or geochemistry. They've never attempted to try and say, what are the impacts on biology, or can we use biology to test whether it's one model or the other? And I'm trying to say, well, we've got the ancient DNA, we can put that on your sample and try and see whether I've got freshwater microorganisms or salt. And that's one of your models or the other one. And we need to be doing more of that big picture type stuff, I think, than the small scale, is it a bear, is it a bison type localized studies. Thank you so much for hanging around. You've done amazingly well.